and subscribe in our YouTube channel. If you have any question, please put your question in the Q&A box, and also please participate in the poll, and this really helps us to improve the quality of our e-seminar series. So our next speaker is Dr. Leila Pirhaji. She's talking about AI-driven drug discovery from invention to commercialization. Same time on Wednesdays at noon Eastern time, we will have our e-seminar series. As always, you can to get the most updated information about our upcoming e-seminars, you can follow us on the Twitter, and you can, if you have any question or concern, you can also contact the organizer, Dr. Akbari, or myself, or our coordinator, Mr. Karamzadeh. These e-seminars are not possible without the support of our sponsors. We would like to thank the Montreal Transmedtech Institute for sponsoring these e-seminars. Montreal Transmedtech Institute aims to support the development of innovative medical technologies, train the next generation of professionals, and make innovations in life sciences and engineering a source of wealth for society. Based on a living lab approach, Montreal Transmedtech Institute provides an integrated environment that supports interdisciplinary collaborative processes and co-creation of new medical technologies and interventions to catalyze their development and adoption by users. And also, we would like to thank our other sponsors, University of Victoria, University of Victoria's Research Partnership and Knowledge Mobilization team facilitates the development of research partnerships, commercialization of university-developed technologies, and support for new startups. By that, I would like to introduce our speaker today, Professor Shana Kelley. It's a great honor to have her here with us today. Dr. Kelley is a university professor at the University of Toronto and a member of the Departments of Chemistry and Biochemistry, Pharmaceutical Sciences, and Biomedical Engineering. The Kelley Research Group works in a variety of areas spanning biophysical, bioanalytical chemistry, chemical biology, and nanotechnology, and has pioneered new methods for tracking molecular and cellular analytes with unprecedented sensitivity. Dr. Kelley's work has been recognized with a variety of distinctions, including being named one of the Canada's top 40 under 40, NSERC, EWR Flow, the 2011 STICI Prize, and the 2016 NSERC Brackhouse Prize. She has also been recognized with the ACS Inorganic Nanoscience Award, the Pittsburgh Conference Achievement Award, and Alfred P. Sloan Research Fellowship, a Camille Dreyfus Teacher Scholar Award, an SF Carrier Award, a Dreyfus New Faculty Award, and was also named a Top 100 Innovator by MIT's Technology Review. Dr. Kelley is an inventor on over 50 patents issued worldwide. She is the founder of 3 molecular diagnostics companies, Genome Sciences, acquired by Becton Dickinson in 2005, Zanigenic, and acquired by General Atomics in 2017, and Cellular Analytics. Dr. Kelley is the director of the PRIME, an initiative focused on next-generation precision medicine that unites physical scientists, engineers, biologists, and clinicians developing new therapeutics and diagnostics. Dr. Kelley serves as a board of director for the Fight Against Cancer Innovation Trust and the Canadian Academy of Health Sciences. She is an associate editor for ACS Sensors and an editorial advisory board member for the Journal of American Chemical Society, Nanoletters, and ACS Nano. This can go on and on. And I would like to introduce to invite Dr. Kelley to share her screen and talk about her journey.
All right, thank you very much. Can you see my screen and hear me okay?
I have to unshare probably my screen.
Let me
know. So, yeah, so can you please share? Yes, we do see, and exactly.
Okay, how does that look now?
It's great, fantastic. Thank you.
Okay. All right, well, thank you so much for having me here today. I really want to thank our organizers. I think it's so important for us to stay connected during this time. And by having events like this, it really brings our community together and keeps us focused on very positive things like the science that we're all doing. And so I really appreciate being included in this series, and I'm really excited to be here today to share my work and some of the work that we've done on commercializing the research that we've done at the University of Toronto. And my group in general works on a variety of really sensing platforms. Over the years, we've developed sensors where the applications are more focused on intracellular sensing or in situ sensing on surfaces. We have technologies that we've developed for rare cell profiling for things like liquid biopsy and high throughput genomics. But the project that I'm really going to focus in on today is a class of sensors that we've developed for in vitro biomolecular analysis, meaning we're going to be analyzing clinical specimens or clinical material in order to provide new solutions for clinical diagnostics. And really the rationale for this work, and this is a slide that I've used for, I don't know, 5 or 6 years, which in current times seems very relevant. You know, our focus over the years has been on infectious disease diagnosis, just acknowledging that there could be a situation coming down the road like the one that we're currently in, where it's absolutely critical that we're able to limit the spread of viral pathogens, be able to manage pandemics more successfully, be able to identify new viral strains, new viral pathogens, also be able to very rapidly identify infections so that they can be treated properly, and limiting the spread of antibiotic resistance. So these are problems that we've been thinking about for some time and really trying to bring new solutions, new tools, new diagnostic platforms so that the diagnosis of infectious disease is much more efficient. And from my perspective, I've always thought that the way to do this is to develop sensors that would be able to read out the presence of bacterial pathogens, viral pathogens, fungal pathogens, you know, any type of microbe that we may need to know about in our environment, in our bodies, in clinical samples. And if you look at the number of different types of sensors that are out there, I mean, many of the technological innovations that we've seen over the last 5 years or so are really driven by sensors, right? Self-driving cars could not do what they're able to do without a massive number of sensors. And so while we have sensors for a lot of mechanical phenomena and optical phenomena, it's really been much more difficult to produce sensors for biomolecular species. And this relates to a number of different issues, but the main one is just the difficulty of being able to get biomolecules or viral particles or bacterial cells to bind to a surface and behave in a manner on a surface that they would, let's say, free in solution. And some of the issues relate to the challenges of achieving high levels of sensitivity, so a low level of false negatives, a high level of specificity, which is a low level of false positives, and also being able to provide a readout very rapidly. And over the years, some of the parameters that we've really focused on in trying to drive the performance of sensors for infectious disease and other applications has really prompted us to look at the specifics of molecular complexation on the surface of a sensor, both from the perspective of kinetics, but also thermodynamics and how they're perturbed on surfaces, and also to pay close attention to how diffusion through a sample may be limited by the size of a molecule, it may be limited by the type of medium that's present. And so these types of parameters have really limited our ability to produce sensors that have clinically relevant performance. But what I'm going to do today is to take you through a series of systems that we've worked on where we've looked at these parameters, we've tried to kind of find workarounds for some of the things that are more challenging. And then I'll describe to you how eventually we have also been able to commercialize one of our platforms and how we have another that we're ready to kind of start moving ahead with. And so the talk, I'll kind of divide it into three parts. I'll tell you about a class of high performance sensors that we've developed that are made out of nanostructured materials. They're electrodeposited metals. I'll tell you about the applications that we've developed for these sensors. And then at the end, I'll take you through a new system where we're really doing what I call striving for simplicity, really trying to make sensors simpler and simpler to embed in commercial products or clinical diagnostic platforms. And so in this area, we have a new solution for reagentless sensing. So, and I'll make sure to keep this so that we're done by 12:45 or so there's plenty of time for questions. And so the system that I'll start with is a class of chip-based detectors that we developed to allow us to look in clinical specimens for molecular analytes. These are three-dimensional sensors that have a lot of surface area, but also a lot of nanostructuring to really promote efficient kinetics and thermodynamics for biomolecular complexation taking place on the surface of these sensors. But before I jump into exactly how we make these sensors and how they work, I'm just going to tell you a bit about some of the foundational work that we did that really motivated us to be looking at these types of three-dimensional architectures. And to tell you about the foundational work, I'll rewind quite a bit and tell you about some of the work that I did as an assistant professor in Boston. And our goal here was to develop readout systems for things like nucleic acid molecules, let's say part of a viral genome or a bacterial genome, and to be able to do that on the surface of an electrode so that we could use electrochemical sensing, which is very cost effective to make into a piece of instrumentation. But the challenge we knew would be to be able to look at the very low levels of nucleic acid sequences that you would find in a clinical sample. Usually in clinical diagnostics, the way this is done is by using the polymerase chain reaction to amplify levels of a target sequence. But it turns out this type of enzymatic amplification it's really difficult to automate. It's very difficult to scale. We're seeing this right now with the limited levels of testing that we're able to do for SARS-CoV-2. And so we thought maybe by building electrochemical amplification into a detection system that we would use with sensors, that maybe this would allow us to circumvent the need for enzymatic amplification. So the way this works is sketched out here. So we have an electrode surface. We have a piece of nucleic acid that we've immobilized. This is going to be our probe that's going to bind to the sequence that we're looking for in a sample. It turns out we use a neutral type of probe. It's peptide nucleic acid, so that the charge is 0 as we start. And then we're going to be looking for the binding of this incoming sequence, which again could be a snippet of a viral genome, a bacterial genome. And when that sequence binds to the probe molecule on our electrode surface, it's going to bring with it quite a bit of negative charge and change the overall charge of this complex that's immobilized on the surface. To read out this change in charge, we use a combination of 2 redox active ions. One is ruthenium hexamine, which is positively charged, so it's going to bind to this negatively charged monolayer. And then we also use ferricyanide. And so the way that this works is that we put a potential on the electrode, we send the electrons to the rutheniums that are binding to the monolayer at levels that correspond to how much of that target strand is bound. But we include the ferricyanide so that the electrons can keep on going, so they can be passed from the ruthenium ions to the ferricyanide ions. And so then there's catalytic turnover that happens that amplifies the signal that we're able to obtain quite significantly. So this is how we thought we might get around enzymatic amplification. But it turned out, after we really tested this approach and put it through the paces with different types of samples, that we were not able to get that much sensitivity. And we were really stuck in the low micromolar or high nanomolar concentration range, which was not going to allow us to look at clinical samples. Luckily, and really serendipitously, we happened to be collaborating with a group that had developed these gold nanowires. These are basically wires that you can generate using electrolysis deposition in a polycarbonate membrane. You can then use a plasma treatment to expose the wires on the surface of the membrane, and then you can clip in and make it an electrode kind of on the backside of the membrane. And these 3-dimensional wires, about 10 nanometers across, when used with this electrocatalytic detection assay, actually increased our sensitivity by 10 orders of magnitude. So without doing anything else to our system, just changing out the electrode, we were able to really enhance detection limits and bring them into a clinically relevant range. And so this was really a breakthrough moment for us in terms of achieving performance that would be much more relevant for clinical diagnostics. And I'll talk a bit more about this, but the way that, or the reason that we think these nanowires work so well is that the fact that they're 3-dimensional really promotes biomolecular complexation, because the probes, when they're displayed on the wires, act much more like they would in solution. They have a higher binding affinity, they exhibit faster complexation with incoming target molecules, and I'll tell you a bit more about this effect as I go. And so we did a lot with these nanowires, but as we started to think about transitioning into a type of electrode platform that we could commercialize and scale up, we decided to move away from polycarbonate and try to get on a chip-based platform, again, to really help us with scaled-up fabrication. So we teamed up with Ted Sargent, who's an electrical engineer at the University of Toronto, and with his group developed a process using just very conventional photolithography to generate the chips shown here. So these are fabricated on a silicon wafer or a glass wafer. We pattern an array of contacts. So this is where we're going to clip onto the chip to apply a potential and measure a current. We have these leads that are going to separate our sensors from the contacts. And then we cover the entire surface of the chip with a layer of silicon dioxide. So that's going to passivate the surface of the chip. And then we open up these very small openings at the tip of each lead. And this is where we thought we could deposit nanostructured materials to try to recapitulate the really nice performance that we got out of our nanowires, but again, in a chip-based platform that would be much easier to scale. And so we learned how to do this using either electrolysis deposition of metals or electrode deposition. And what happens is that as you plate the metals into the pore, they fill up the pore and then spill over on the surface of the chip. And you do get this really nice nanostructuring on the surfaces. So the sensors are a few microns across, but they have a high level of nanostructuring on their surfaces. Eventually, we went over to palladium electrode deposition because this gave us really nice structures where we could really very precisely control the surface morphology. And we could play with all the parameters that you're seeing here to change the size of the sensor or change the surface roughness. The most effective parameter that allowed us to really change the level of nanostructuring on the surface was the plating potential. So if you look at this row of sensors here, this one was plated at a very low potential. And so the structure was built very slowly. All the voids in the structure were built in, and it's essentially a hemisphere of palladium. As we make the electrode deposition go faster, you can see that the surfaces are rougher because we have voids that are forming in the microelectrode then that are not filled in. So with these types of structures in hand, we were able to normalize for surface area effects and then really do an apples to apples comparison study to see which sensors performed the best. And what we found was that it was really this highly nanostructured sensor that had feature sizes on the order of about 10 nanometers, the same as our templated nanowires. And these structures gave us detection limits in the attomolar range using that same electrocatalytic detection strategy that I told you about. Whereas a sensor with more moderate levels of nanostructuring in red here gave us femtomolar detection limits, whereas the totally smooth sensor gave us detection limits in the picomolar concentration range. So we were able to show that there was a direct correlation between surface nanostructuring and detection sensitivity, and with our best sensors could see about 100 molecules of a sequence in a sample. And we've done lots of studies over the years to really understand why this is. We've done lots of experimental studies that I think are compelling and have taught us a lot about how these types of systems act. But what I want to show you is a set of molecular dynamics simulations that I think really drives home the point. So here what we did in collaboration with a group at IBM was we created nanostructured surfaces with probe molecules immobilized on them, or flat surfaces with those same probes attached through a thiol linkage. And then what we did is we ran the simulation. It took place over a time window of about 40 nanoseconds. And then we looked at the dynamics of the probes. We looked at intermolecular interactions and intramolecular interactions to see how the probes were behaving on these different types of surfaces. So here at the top, hopefully you can see this. This is the simulation running. And so you're seeing the probe sample the water molecules and move around on the surface. And so what we could do is categorize the behavior of the probe according to whether the probes were participating in intermolecular aggregates, so interacting with each other, intramolecular aggregates where they're interacting with themselves, or if they were in a nice free elongated conformation. And it's this free conformation that we think then would make the sensor and the probe more likely to interact with a target molecule. And so running this simulation, we did this in the presence of a variety of different cations just to see how that would perturb the simulation. And you can see that on a flat electrode, as you increase the valency of a cation that's in solution, you introduce the amount of intermolecular aggregation. If we instead run the simulation on a nanostructured surface, you can see the probes are much more resistant to aggregation. And so again, we think this comes back to the display of the probes on the surface of the nanostructured sensors and the fact that they're really much more in this free conformation that allows them to interact with the target molecules that we're hoping to detect. And we think that that's really where the sensitivity comes from. So with this system in hand and performance that we thought was clinically relevant, we then went on to start looking at different clinical indications. And so you can look through the literature and look at what the loadings of bacteria would be, let's say, for a patient that had a blood infection, or if you were to swab someone to look for MRSA colonization, or which is a pathogen that you very often find in the nose. or looking at STDs or UTIs, you can see that you have a range of concentrations of loading that you would have to work with in a different type of clinical sample. And then if you take those numbers and then turn them into concentrations, assuming that you're going to be looking at, let's say, a messenger RNA because it's single-stranded, so a little bit easier to look for. You can multiply by 1,000 because there are usually multiple copies of messenger RNAs, and so you can get the concentrations that are listed on the right. So you can see that what's required is that we have somewhere between attomolar and femtomolar sensitivity in our system. And we realized that in starting to look at these applications and also in thinking about looking at messenger RNAs, that we also needed to consider the length of the molecules that we were looking at. To kind of develop the system, we were only looking at short oligonucleotides. Now we're talking about messenger RNAs, which are much, much longer. And so we ended up running a series of simulations just to look at this. And what we found is that while a 20 base oligonucleotide, if you look at sensors that have a variety of different diameters, the time that is going to be required to accumulate a single molecule on these sensors is pretty short. And so if you're interested in rapid turnaround, which is really important for infectious disease diagnosis, even a 10 micron sensor is going to give you good accumulation levels of that analyte. But if you look at a 4,000 base messenger RNA, you can see that the situation is much different and that you really need much, much larger sensors in order to have a chance of that molecule landing on the surface. It's just like jumping out of a plane with a parachute and trying to land on a football field versus a particular blade of grass, right? You'd have a much better chance of landing on the football field. So we realized that the size was really important and that this few micron diameter that we had been working with probably wasn't enough. So we revisited the materials chemistry. It turned out that palladium, while it will give us a footprint of about 100 microns across, it wouldn't give us any verticality just because of the way that palladium electrodeposits. But if we used gold and we used very rapid growth conditions, we were able to produce these nanoneedles. And these needles then gave us a nice large structure that really satisfied the parameters and the footprint requirements that our model had told us about. And these structures are really interesting. I won't dwell on the materials chemistry today because I want to tell you about kind of where we've taken these systems. But in some work done by Sarah Mashid, who's now on the faculty of McGill, in our first couple of years there, she did a very beautiful and a very careful study of these nanoneedle-based electrodes and was able to see that every single needle in the structure had 5 sides. And she was able to show through a Fib experiment that that was due to 5-fold twinning within the needles that then really drove that as a structural feature. And it turns out that these pentagonal needles are growth hotspots when you're doing electrodeposition under really fast kinetic conditions. So with these structures in hand, and this was the work of a graduate student in the group, Ji-Chao Feng, who's now at Bristol-Myers Squibb, but also Leila Soleimani, who also did a lot of the foundational work that I just described to you, who's now a professor at McMaster University. They were able to show that with these structures that in solutions or samples that contained about 1 cell per microliter of bacteria, that we were able to read out the presence of that bacterial pathogen in about 20 minutes. So really giving us the speed required for point of care detection. And so I'll tell you now just a little bit about the commercialization path for this technology. We started a company called Exogenic, spun it out of the lab. The idea behind the company was to take this sensing technology, build a disposable cartridge around it that would include not only our sensing chip, but also the reagents, so that ruthenium hexamine reporter I told you about, ferricyanide, buffer, and the cartridge would have fluidics and everything that it would take to fully automate this assay. And so that disposable consumable was developed, and also a benchtop analyzer with the system that was the final product being able to take a swab specimen. That swab would go into a tube would go into cartridge would go into the instrument, the user would hit go and then get a yes/no answer back. And the company was able to do this at a pretty low price point, both for the cartridge and the instrument. And so they were successful in automating this and showing the clinical utility. But I'll tell you a little bit about some of the ups and downs along the way. So this company was started in 2010, and the little picture there just shows that what we started with was a really crude prototype, which was just like an electrochemical cell on the benchtop. And then it took us about a year or two to produce the first fully automated instrument. We then moved to a beta prototype, which was a more refined instrument. But really where a lot of the work had to take place was in scaling up manufacturing. And this was really a challenge, and I apologize, my PowerPoint's kind of doing the animations for me. The manufacturing scale up was one of the longest parts of the process, because we had to build a manufacturing facility to produce the cartridges so that we would have enough to do a clinical trial. And there were a lot of processes that needed to be worked out to actually manufacture that cartridge. But eventually we were successful in doing that and developing a clinical grade system that would be compliant with all of the regulatory requirements that you need to satisfy in order to test patient specimens. And so we did eventually go out. We did a beta study at a local STD clinic. The lead product was an STD test, and successfully showed that the system could produce results that were comparable with the lab-based test that was used for the same indication. And then the other challenge that we ran into that in the end had us sell the company to a US defense contractor, General Atomics, was that we needed to scale further. So in order to have a commercial product, we needed to produce not thousands of cartridges the way that we did for the clinical studies, but tens of thousands, hundreds of thousands, eventually 1 million. And to do that, we really needed a partner that had the resources to be able to continue to scale. General Atomics was a bit of an unlikely partner for us. As I said, they're a U.S. defense contractor best known for producing the Predator drone that was originally used to do aerial surveillance in the Middle East. But they've also kind of started focusing on more peaceful fields and have started a molecular diagnostics division, and so use this system to really kick off that effort, and are deploying the technology now in the U.S. military. So we're very excited to have seen this go all the way, from bench, from very early exploratory experiments, all the way to a product that can now be used for clinical testing. So I've told you about the infectious disease applications of this technology. Over the years, we've done other things in the lab, including using it to allow surgeons to assess organs that are under evaluation for transplant. We've made some systems for bioproduction of therapeutic cell-based therapies. We have sensors for drug monitoring and liquid biopsy. So we've done a variety of things with this one platform. In the last 10 minutes or so, I want to tell you, though, about a new sensing system that we developed really in response to everything that we learned through developing the sensors that I just told you about. And as powerful as this system was, I mean, just watching the process that was required to automate and taking a sensor and taking the reagents and having to produce the reagents, produce the sensor chips, embed them in a fluidic system that would deliver all of the fluids. It is really complex, even with a relatively simple system like the one I just showed you. And so I had been thinking about this for some time. I'll also tell just a quick story that I think will resonate with the Canadians that are part of this event today. I ended up being on a show that it's not on anymore, but used to be an interview show, kind of like 60 Minutes for people from the States. But this show was called Mansbridge One-on-One. It was hosted by one of the premier journalists, television journalist, Peter Mansbridge in Canada. And he had me on his show to talk about new diagnostic tools. And he asked me during the show, he said, Shana, I have a Ford pickup truck. And anytime I need to take my pickup truck into the shop, a sensor goes off and tells me to do that. Why don't we have these kinds of sensors that monitor our health? And I didn't have a great answer for him, except it's really complicated, Peter. So in not having a good answer for that and all the experiences that I just told you about, it really pushed me to start thinking about even simpler systems that would hopefully get us to a point where we would have just sensors, autonomous standalone sensors that would alert us to problems with our health, the presence of pathogens in the environment or our bodies, without the need for really complicated fluidics and all of the other things I've just told you about. So, there are examples of what we call reagentless sensors out there. And this idea of being reagentless or reagent free is very attractive because it takes a lot of the complexity out of it, right? If you don't need reagents, then you don't need to embed those reagents in a cartridge. You may not need the fluidics. And it's just a much simpler way to produce the type of sensor that I've always thought would be very powerful. And, glucose sensors are a great example of a reagentless sensor, right? They're just sitting there waiting to measure the presence of glucose. There are examples of electrochemical sensors that have been developed to look at neurotransmitters directly in the brain, but there you're reliant on having chemistry of that neurotransmitter that is redox active. And then there's also the beautiful work of Kevin Plaxco and his coworkers that have developed these aptamer-based eDNA sensors that change conformation and can read out the presence of analytes without reagents, but they are limited to aptamers. And so we were looking to develop something that maybe would be more universal, more amenable to direct detection of, let's say, viral pathogens, proteins, and really any analyte of interest. And so I'll describe this concept to you. So what we started with was the idea of trying to track the motion of a molecular pendulum. And so the idea here is that we have a pendulum, and I'll tell you about the molecular structure in a moment, but that pendulum is going to be moving on a surface, and that movement and its diffusional kinetics should be connected to the size of the bob on the pendulum. And we wondered if this would be a way to start reading out the complexation state, let's say if the bob of the pendulum was something like an antibody. And we thought we could control the motion of the pendulum using an electric field that we could generate on the surface of a sensor. If we used a linker for the pendulum that was negatively charged, we thought if we induced a positive charge on the surface, we could then bring the pendulum to the surface with kind of a defined kinetic profile. And you can model this out, and I won't take you through all the physics, but basically you have to think about the drag on the pendulum and how that's going to affect transport, and then also the impact of the electric field. And so Sarath Gomes in the group, who's a really talented graduate student in our electrical and computer engineering program, did all of these simulations to show that depending on the length of the linker, the charge of the linker, and the applied potential, that you would see these different diffusional profiles with this tau parameter that's on the y-axis of this plot, just being the characteristic time that it would take to transport this molecular pendulum to the surface. He also showed that the diffusion coefficient of the bob of the pendulum would also modulate the kinetics. And in the end, he was able to do simulations of what an electrochemical experiment might look like if we applied that potential to the surface, had the pendulum come down, and then look at the decay of the current, which is telling us about the motion of the pendulum. And what he was able to demonstrate is that if we, let's say, had a pendulum and then we bound something to it that was on the order of 25 kilodaltons, that we would see a pretty dramatic modulation of this current transient. And so those were the simulations. So here I'm showing you what the molecular pendulum is actually composed of. So we use DNA as our linker. It's negatively charged. As you know, it's nice and rigid, so it's a really good linker. We use an antibody as the bob of the pendulum, and this is what's going to recognize a specific analyte. which is going to bind then to the antibody. And then to read out this electrochemical process, we use ferrocene, so just a small iron complex that we attach to the DNA structure. And so again, the idea is that we start off with this sensor. It has everything that it needs to read out the presence of an analyte. And so we shouldn't need any other reagents. And then in the absence of the analyte, the travel to the surface of the sensor or surface of the electrode is going to be faster because of the decreased drag force relative to a sensor that has the protein bound, that then is going to be slowed down by the additional drag on that bound protein. And so we thought we would be able to read this out looking at electron transfer kinetics, which you can do using chronoamperometry. And so our first model analyte was troponin. So this is a cardiac biomarker. It has a molecular weight of about 24 kilodaltons, a hydrodynamic radius or diameter of about 4 nanometers, and an additional charge of +2, which will affect the transport given that it is sensitive to charge. And so sure enough, if we do the chronoamperometry experiments where we pulse a potential, we measure the decay of the current, we can see this change in decay and a change in current that was described by Seurat's model. And the experimental side of this was done by Jagannathan Das, who's a research associate in the group, who actually came up with the idea of this molecular pendulum as a way to do reagent-free sensing. And so you can use this to do continuous monitoring. So just taking a sensor, dipping it into a solution, having an analyte be added to the solution, and then watching that current transient change. It's also the sensors are also very stable. If you bring an analyte in, take it away, bring it in, take it away, then the sensor responds to that. So it's a reversible process to have analytes bind, unbind to the sensor to allow us to make dynamic measurements. And so we've done this in a variety of different biofluids, saliva, sweat, tears, urine, blood. It's compatible with all of these different types of biofluids. And then we've also looked at a variety of different proteins that are markers for cardiovascular disease, markers of inflammation, allergy, stress. And so it is a general method. And then we've also been able to make these measurements in vivo. So taking a very small electrode array, putting it into the mouth of a mouse, and if you've never looked that closely at the mouth of a mouse, it's very, very small. So we had to really miniaturize our electrodes. And what we did first was to look at a model of heart failure. And so we did this by treating mice with doxorubicin and then collecting or doing in situ measurements of their saliva to look for troponin as a marker of the cardiac stress that the doxorubicin would induce. And so you can see here that as we treat the animals and we look at the samples either in vitro, so we take the saliva and analyze it, or we put the sensors in the mouth of the mouse, we can see the rise in troponin levels. We can also do measurements where we inject troponin directly into the mice and then watch for it to come into the saliva. And so this is a continuous measurement, so we just leave the sensor there, and then we can watch the signal come up as the troponin levels rise in these animals. And so we're very excited about the performance of the system and the applications that it opens. And we really envision developing a variety of different sensors where continuous real-time monitoring of physiology would be relevant. And then just to finish, I'll quickly tell you about how we have adapted this for SARS-CoV-2 detection. Obviously, when the pandemic really took off in Canada, all of us were thinking about how our science could potentially be repositioned to have an impact on the pandemic. And it was noted very early on that in addition to being able to see the virus on a nasopharyngeal swab, that it could also be detected in the saliva. And so just thinking about the fact that we had these sensors that worked really well in saliva and that the virus seemed to be in the saliva, we wondered if we could make devices that would allow us to, within minutes, assess whether someone was carrying the virus in their mouth. And we thought this could be an alternative to the relatively slow PCR-based testing that's done in labs that's also very bottlenecked. And so again, the idea here was to be able to look at the difference between one of these molecular sensors uncomplexed on a surface versus bound to a viral particle. And this illustration is to scale. It shows you how enormous the viral particles are relative to our little sensors. And so accordingly, when we look at electrochemical traces, again, using chronoamperometry, we see a huge modulation of the current. And we've shown that we can do this in saliva, and we're now doing animal studies to show that we can do direct measurements in the mouth. And so this is work that's ongoing. We're developing instrumentation. We have a kind of a first generation version of a handheld prototype. We're doing lots of clinical sample testing, animal models, as I mentioned, and we've also formed a new company, Arma Biosciences, to hopefully then take these prototypes and enhance them and really take this technology, which we think has tremendous applications for physiological monitoring above and beyond SARS-CoV-2, to develop them and commercialize them. So with that, I want to summarize. Hopefully, I've convinced you that these types of electrochemical sensors have a variety of applications for biomolecular analysis in the management of infectious disease. Of course, we've worked in many other areas. And our work now has really shifted to this reagent-free type of platform, just in the hope that it will really kind of finally enable us to make sensors that can be used much more widely to monitor physiological analytes. So with that, I want to thank my group, everybody here. This was a picture taken, of course, before we had to stay much more distance. This was last fall. But I really thank everybody who's a member of the Kelley Group for all of their creativity and hard work. In particular, I just wanted to mention Jagadamoy Das, who is the research associate who developed our reagent-free sensing system, Sarath Gomez, who did the simulations and has also been doing all of our prototyping, Hanya Yousefi, who has adapted the system for SARS-CoV-2 detection and was really instrumental in getting that project off the ground. I thank all of our collaborators. I thank our funders. I thank you very much for tuning in today. And I'd really be delighted to take your questions.
Well, thank you very much, Shana, for your very interesting and inspiring talk. So I think if you unshare your slides, you will be able to see us. I know that you've been only talking to your monitor, to your computer, but now we can communicate. So if you have any questions for participants, please make sure that you ask your questions in the Q&A box. So we have a few questions now. But before we start the Q&A session, I'd like to go over a few, again, remind you about a few points. First one is that next week, our speaker is going to be Dr. Leila Pirhaji, who is going to talk about artificial intelligence and its application for drug discovery. We're looking forward to her talk. This is the first AI-based talk that we're going to have next week. We also would like to thank our sponsors, Transmed Tech Institute, which and also University of Victoria. Transmed Tech aims to support the development of innovative medical technologies and train the next generation of professionals and make innovation in life sciences and engineering a source of wealth for society. Transmed Tech is based on using a living lab approach to provide an integrated environment that supports interdisciplinary and collaborative basically processes and co-creation of new medical technologies and interventions to develop new technologies. We also would like to thank our other sponsor, University of Victoria, Research Partnerships and Knowledge Mobilization. UVic's knowledge mobilization team facilitates the development of research partnership, commercialization, and university-developed technologies, and then also supports the new startups. So with that, now let's start the Q&A session. Again, it was an amazing talk. I learned a lot from you, Shana. What a great technology that you developed, and then what a great story. I mean, amazing. How you start from basic science and then push things forward towards a device or a technology that is ready to be commercialized and that is ready to be used by the end users. Especially during the pandemic, we now appreciate the work that your team, and then of course other teams who are developing sensors are doing more. So thank you very much for saving us. So let's start with the questions from the audience. So the first question is from Rasul Sayed Mahmoud. So he asks, his question is about using smart lenses, and then the potential of using smart lenses and combining it probably with your technology for diagnostic applications.
Sure. I'm assuming that smart lens meaning like Google glasses, or yeah, okay. It's always hard to
things like this.
This way. Yeah, I mean,
no, smart lenses are, Shana. Smart lenses are those that I mean you put in the eye. There are like these lenses that have electrode.
Right. Absolutely. Yeah. So like contact lenses. Absolutely.
Contact
lenses.
That's
something. Yeah. Exactly. So that's something that we're looking at, because that's perfect for us to be in contact with a biological fluid is exactly where we need to be. And so we've done a lot of our work in saliva to begin with, just because it's just easier. We can take one of our existing sensors and just put it in the mouth. But the lenses are very attractive. And I know there's people in my group that are really interested. Building kind of on-device electronics is not as much our expertise, so we haven't done it yet. We would love to team up with somebody that was their expertise. So I'd love to hear from people that know how to build those things.
That's great. It's really cool. And then I think there is potential to combine this technology with implantable sensors and then microneedle technologies and so on and so forth. I think this could be another cool application for your technology. So the other question is from Mark Loss from Biotechnology Center in Gliwice in Poland. So the question is about in vivo nanosensing, and then Mark asks that these types of nanosensors, when you implant them to measure biomolecules in vivo, they're usually associated with the buildup of fibrosis around the sensors. I mean, in glucose, for example, sensors. So could you discuss the issue briefly and perhaps offer remedies?
Yeah, that's a great point. I think for the applications that we are envisioning, we'd probably just like to stay out of situations where you're implanted to a point that fibrosis would occur. I really like the idea of saliva monitoring. I mean, you can put a device on a dental implant. You can have it come in and out. You can clean it. It's not going to perturb the tissue. You of course have to be working with analytes where that's going to give you meaningful information. We are also now starting to look at interstitial fluid monitoring, where that may be more of an issue. I think the idea of being in the eye, on the surface of the eye is another very attractive one. Urine testing. So, I think with in vivo sensing, unless you absolutely need to be embedded to the point where you would get fibrosis, there's a lot of other things for us to do. And it's not really our expertise. Our expertise is on the sensing part of it, not necessarily getting around that. So I think we'll kind of stay away from that by being in other parts of the body.
Okay, great. So I have one question along the same direction. Now that you mentioned that you're interested in using the sensors, especially in the for saliva testing, is that the sensors that you're developing are on rigid bodies, rigid substrates, not rigid bodies, on rigid substrates. So, but when you want to put them in, like in the mouth, let's say, for continuous monitoring, because eventually that's, I think, the second project is moving forward, or maybe on a wound, probably, that you want to measure the markers at the wound site and probably see if the wound is healing or not, or if there's any infection at the wound site. So, have you thought about using this technology and then bring it on flexible substrates that conform to irregular shapes that we have in the body?
Yeah, absolutely. That's a very important direction. We don't necessarily need to be on a rigid substrate. It's just that the photolithography we use needs a fairly smooth surface. What's critical for us is having a good electrode, though. Mm-hmm. And gold is for what we do the best electrode. It just the kinetics are very fast. It's very clean. It allows you to see small changes. And so going over to something flexible, we have to find a way to create a nice gold electrode on whatever material it is. Or we can use other electrode materials. Just gold is the one of choice. And so that's something that we're working on. What we found with some commercial systems is very often, you'll have chromium in there or copper or other metals that really mess with electrochemistry. Mm-hmm. So that's something that we're in the midst of working out. How can we get on to other substrates? Because as you mentioned, for physiological monitoring, you really don't, even putting a piece of glass in somebody's mouth, that's not a very good thing to do, right? So we need to be in other types of systems, and we can be on just a piece of wire. We don't have to be on a substrate. We can have a little bundle of electrodes as wires. And so I think we'll get there. It's just the materials side of things that has to come along.
For sure. And then it's a highly interdisciplinary field and then project. So it has many different. All the projects I see that have many different needs, different expertise, and different people from different backgrounds to help with the development of the technology. So, but besides this. These scientific questions that we had, I'm interested to know more about your path towards commercialization, and in particular, I'm interested to know about your interactions with the FDA. This is something that is very critical in terms of developing technologies and bringing them into market. So, what was your experience? Have you gone through the FDA or regulatory approval process yet? And then, if so, what were the challenges and your experience? I would like to hear about that.
Yeah, so I have been involved with parts of the process that's required to get regulatory approval, and I think the thing that's very unexpected is that agencies, the FDA and Health Canada are actually very friendly agencies. They want to help. They're not there to impede you. They are there to help you. And you can find people to talk to, and they will give you guidance, and it's very good information. The challenge is that it's very complicated. The quality side of things, it's not even just the regulatory, it's the quality management side of producing and manufacturing a device that's going to be used to produce very important information. And you have to have, so I'm sure you're aware, volumes and volumes of documentation about exactly how the thing is made, how you're making sure that nothing is drifting, everything's controlled, all of your lots of consumables are acting the same way. I mean, it just goes on and on and on. And that's why, starting companies in this area is not like starting a software company, right?
Yeah.
I mean, it's not just a thing that you develop and then you make it run, and as long as it works, it's okay. You have to, and so you need deep expertise in regulatory, in quality, in product development, which is not what most of us think it is. I mean, it's a very rigorous discipline that requires incredible expertise. So this is I think one of the biggest challenges, that these types of sensors, these types of testing systems, which do not get reimbursed at a high level, they cost a lot of money to develop because of all of this. So, but we need them, obviously.
Yeah,
We've
we
got
need them, and I agree. And then one thing about QMS, quality management system, that I'm sure you have put in place right now for your companies to go through the regulatory approval. So my question is about whether you are implementing the same process in your lab, in your research lab, because you have two hats, right? A professor hat and then an entrepreneur hat. So are you using these in your lab to just for the future technologies that you're developing? So everyone is like following these processes,
Yeah, it's hard because part of the quality management is locking everything down and never changing it. And that's not what we do in the lab. We change things every day, right? You come into the lab to change 5 more variables and see if you can get this working or understand the behavior. And so you really can't use it. It would you stop yourself from making any progress. But what you can do, and I'm sure you guys do this, and a lot of people who are listening, develop SOPs, standing up, put it on paper. How do you do this assay? How do you make your buffer? Anything that is locked should go on a piece of paper so that you can help train new people. You can help make sure that everybody's doing something the same way so that you can explain any differences in the results that you're getting. So I think that aspect of it is really important, but the lockdown, you can't change anything, right? That works for development. It doesn't really work for research.
I see. Great advice, because we are developing a smart bandage technology, and then we have our startup, and then we are trying to learn and then practice this quality management process. And then I was trying to implement it in the lab, but I realized that it's very difficult because of the reasons that you mentioned. That's great to hear that. I mean, it's a little bit of relief for me. So for the participants, if you have any questions, feel free to ask in the Q&A box. So we have 2 more questions from the participants. The first one is from Gilberto. Amazing talk. That's what Gilberto says. And then the question is, have you thought about working with digital microfluidics or combining your technology with digital microfluidics?
We've thought about it and we've done it. So I'm very fortunate here to have Aaron Wheeler, who is one of the world's experts in digital microfluidics. So Aaron and I have worked together lots over the years. He's one of my favorite colleagues. I mean, we sit on each other's students' committees. We do a lot of things together, and it's wonderful when you're at a university where you have people that are so close to your field. And so we, on the first technology that I told you about with these nanostructured microelectrodes, we were able to embed those in one of Aaron's devices to show that these reagents that I told you about, that you could move them around using digital microfluidics, you could manipulate the sample, you could wash the sensor, you could do everything. It's very cool stuff, as you know. So we have worked on that a bit. And it's challenging because DMF is driven by electrical potentials, right? Very high potentials. And so integrating our little sensors that actually work at very low potentials with this high voltage stuff that they use was actually more of a challenge than we thought it would be, but we did it.
Yeah, I kind of knew the answer because I know Aaron is also so it was kind of interesting to know that. I mean, it's again, I mean, collaboration is very important, and I'm glad that one of us now we hear from experts, board leaders, in the field. On different areas that I mean they collaborate. It's all about collaboration. It's not only my work. It's not only sitting in my silo. It's just collaboration, and collaboration that makes things move forward. And then I'm really happy to hear this from one of the board leaders in biosensing technology. So. So Preeti Sanjay is, the question is about photochemicals. And she says, first of all, she thanks for your great talk. And then she says that I'm working on photochemicals or phytochemicals, sorry. Is that phyto or photochemicals? Please guide me for better, I think it's photochemicals, right? And then please guide me for better advanced research areas in my topic. But I mean, any potential for your technology to be combined with this optical sensors that are also, some of them they go down to single molecule detections, such as whispering gallery mode systems and then other types of sensors. Is there any potential to combine these types of technologies, or maybe like fluorescent-based technologies, and combine them with your sensing technology?
Yeah, absolutely. I mean, the nanostructured sensors that I described, they do have interesting optical qualities. I mean, if you look at the sensors, they're red,
Mm-hmm.
so, plasmonically active. And so I think there are some interesting applications in looking at those structures and using them as service labels. So we've dabbled in that a little bit over the years, but given that we're really, electrochemistry is kind of, it's really the thing we focus on. And the advantages of it that we always talk about are just that you don't need an excitation source, you don't need a detector. And so to bring those things back in, it's not the main area of our focus, but certainly, integrating systems can be very powerful.
Mm-hmm, mm-hmm. Yeah, great, fantastic. So I have another question about, because I see that you are like highlighting some of the like using these sensors for detecting like different molecules in saliva. And then I see, I mean, where you're going. I mean, I understand. I can relate with the reason. I mean, especially with COVID. I mean, no one wants to go through this process of sticking something in their nose or taking a blood sample. Saliva is the easiest way to measure the presence of maybe infectious diseases, or some of them. So, but what are the challenges of detection in saliva as compared to the blood or urine samples?
I think the biggest challenge is just that this is, people's mouths are just not static systems. And we do not yet know what happened. If you just had a peanut butter and jelly sandwich, I have no idea, what our doctor is going to tell you. And so the complexity of what you might find in the mouth, how long does the signal come back after, let's say you drink a full glass of water? We don't know that yet. So I think there's a lot of very practical things that we need to look at. Of course, we're doing our measurements right now on animals. It's a little hard. I mean, we probably could get a mouse to have some peanut butter and jelly, but, we're going to have to find a way to really get at that. So, whereas blood is always, it's blood, like you're not going to have the same kind of complexity, but it's so appealing because it's not invasive.
Yeah.
Right. And, we think about ourselves, like nasopharyngeal swabs and how unpleasant it is, or getting a needle or whatever. But think about somebody with dementia, think about somebody in a long-term care home, and the amount of effort and difficulty that healthcare workers encounter when they're trying to get, a really good specimen of a swab. I mean, it doesn't work. Yeah. And so you need ways that are just much, much less invasive to really enable testing with the people that are most vulnerable.
I totally agree. And again, that's another angle that we're trying to look at these technologies, these especially continuous monitoring technologies that are being developed, and then that reduces the need for elderly or those who live in remote areas to go and then visit doctors regularly. I mean, they can be monitored remotely by the physicians, and then the treatment can be implemented basically again remotely. Great to hear that. I'll have again. I'm sorry that I'm asking so many questions, and then you can stop me, Huma and Shana. I mean, but I'm fascinated with what you're doing, and then my other question is about clinical trials, and then I promise this is going to be my last question. So you have developed this technology in your lab, and then you've done tests in animals, right? And then you got promising results, and then now you want to go into clinical trials. So my last question has two questions. First, when do you decide, or when do you start communicating with your clinical partners? At what stage? And then how do you find? I mean, what is the process of getting funding? How difficult it is to get funding for clinical trial, or how easy it is to get funding for clinical trial? What are the routes? What are the challenges? What are the
These are good questions. So if you have an in vitro diagnostic, right, where you're just going to be taking a sample and analyzing it, it's pretty easy, right? You just need a great clinical collaborator. You need a good protocol. You have to get it approved and research ethics, to bless it. And then you can analyze the sample. So you can do that at any stage, irrespective of the maturity of your technology. If you want to do something that's more of an in vivo measurement with a human, it's a lot more complicated, right? Yeah. And so you have to get the prototype to a point where it's safe, number one, right? And to a point where you can convince people that it's going to collect meaningful information. I mean, a research board will not let you near a human patient until it's clear that you're going to get something out of, exposing somebody to a product that maybe hasn't been through a really rigorous regulatory approval. It can happen, if you have the right clinical partner, it can happen. And then in terms of funding, I mean, I really feel that clinical, like hardcore clinical testing of a new technology is probably best done in a company where you have locked down the quality, you have reproducibility, you're making devices and you know that every single one of them is going to be the same, that the data is going to be reliable. And that's very difficult to do in a university setting. Yeah. So once you're in a company setting, I mean, for the right product with the right profile in terms of being a potential commercial success, venture capital investors will support clinical trials. There's different ways to do it. Our federal funding agencies will support clinical trials. So I think once you're to that point, there's resources out there where I think we have not enough resources and capital in Canada is for the early phase, when you've crossed the line, it's not appropriate anymore for a graduate student to be doing the stuff. It should be, going out to a company, but we don't really have, in the US, there's the SBIR program where you can get seed funding in the form of grants to go into a company just to get that first little bit of work done. And we don't have that. And that makes it really hard, honestly. Yeah. So I hope eventually, we find mechanisms to help people get companies off the ground because startup companies, they're not all going to be successful. It's always the roll of the dice. You got to have the right technology, the right time, the right people, everything going for you. And if you can only start, one at a time, your chance of success is not that great. But if we could get a lot more things out there and seed funded to see whether they have legs, then I think that would be tremendous for our ecosystem.
I agree, and I think that's something that we can discuss with NSERC and even CIHR. NSERC is supporting. I mean, I2I is something that, but compared to SBIR in the States, which the scale is in the order of $1 million, it's a little bit it's good, but it still needs to be improved, especially if we want to. I mean, the Canadian companies want to compete with U.S. companies and even European counterparts, because it's a competitive world, and then we need to be able to develop technologies and then mature them and then bring them into market as soon as possible. It's especially with the COVID situation, we all now it's a competitive field for developing sensors, and that we have to have all the resources available to us in Canada to be able to push things forward and then be the pioneer in the world. Great advice, and then great comments. There is one question from Carolina. So great talk. Have you ever worked in sensors applied to muscle mechanics? So the question is about the application of your sensors for muscle mechanics.
No, I mean, we're really focused on biomolecular analytes more so than kind of force-based sensors. So we haven't, that's just not an application that we've looked at, but maybe we'll get there.
Oh, there are many opportunities. I mean, this is a platform technology, and then you are like, there's a myriad of technologies and opportunities that can be available using this sensor platform. So Human has a question, and then sorry, Shana, I know you are pretty busy. I just want to let you know that we usually go until 10 until 10:30, or it's 1:30 your time. But feel free to let us know if you have any other meetings. Just we can stop.
I can probably go for about 5 more minutes. Yeah.
5 more minutes. That's great. So Human, you have a chance to ask a question. Sorry, and I asked many questions because.
Well, hopefully we'll meet in person sometime soon. We can have
for sure.
Yeah.
Thank you so much, Shana and Mohsen, for a great Q&A session. So I'm just wondering, have you ever tried combining your technology, your sensing technology, with the organ-on-a-chip technology?
We haven't, but that's really interesting, right? If you could embed sensors into an organ-on-a-chip type platform, you could get a lot more information. I mean, a lot of it is done right now optically, right? But if you were able to kind of do real-time continuous monitoring without the limitations that are a problem for fluorescence, that would be very powerful. So, I mean, I'm very excited about this, the idea of reagent-free sensors, because it opens up the ability to do that, right? You're just going to put a little sensor in there and code it with whatever you need to, and then let it do its thing. So I think there's a lot of applications in organ-on-a-chip. There's a huge applications in bioproduction, therapeutic antibodies, cell-based therapies, to have sensors in a culture to tell you, is it done yet? What's it producing? What are the side products? All of these things. And so there's a lot of applications. I mean, we're very excited about it. So that's one, and we have some really great people here in Toronto that do organ-on-a-chip and biology-on-a-chip systems, and so that's definitely on our hit list.
Exactly.
Awesome.
Thank you so much.
Great question. And then
my other. Go ahead.
Go ahead, Human. Oh, okay. So great question. Again, I forgot about organs on a chip. There's a great opportunity for this technology, and especially the continuous monitoring system. I remember when I was doing a postdoc in Ali Khadam Hosseini's lab, I remember that one of the major challenges was to continuously measure the proteins that are secreted from the cells and from these organoids that we were developing in microfluidic devices. And we were always discussing it would be great to have a sensor that cleans itself, and then it provides a available surface for the markers to come into. So thank you, Human, for actually bringing it up.
No problem. No, I'm really interested to combine these two technologies together. As you mentioned, also we have Militra Radzic there that I did my postdoc with her. So I mean, she's expert in this field. So the other question I had, because you have different hats in companies, in university, how do you manage all of these? How do you manage your time, your responsibilities, and how do you recruit great people in your lab and your companies?
These are good questions. In terms of how do you manage your time, I mean, it's a juggling act. We all have juggling acts, right? I mean, if you're teaching, you're running a research group, you just make it work, right? And you figure out what's the highest priority, and then you focus on that. And so that's always what I've done. I think starting companies based on research, right? It's just one seamless continuum, and you have to learn the aspects of commercialization that are not maybe what you learn as a graduate student. I was very fortunate. I started my first company based on a technology that I developed as a graduate student when I was at Caltech, and was part of this amazing journey that was a company that got off the ground in Southern California, that had top tier investors, and I kind of learned the ropes through that. And so then when I wanted to start doing these myself, I just kind of knew, okay, well, you do this, you do that, you can call this kind of investor, that kind of investor, you're do this or that with your IP. And so we figured it out. And I don't know. I mean, I think, as you go, you just get more and more efficient, right? With getting things done. I remember when I had my first child, I thought, how am I going to go from working 12 hours a day, to working how many hours a day I get now that I have this other set of responsibilities. But you just do it, right? And then you get better at doing what you need to in a certain amount of time. So, you have to recognize when you are hitting the limits of your bandwidth, and that can happen. But I don't know. I think these are all good.
That's great.
Thank
That's
you.
great. I have a child too, and then she is sitting there watching TV, and then otherwise she would have been all over my computer. So thank you very much, Shana. It was amazing to talk to you today. I personally really enjoyed your talk, and then I had many ideas in my head when I was listening to your talk. Thank you for sharing your experience with us. Like I'm sure that the audience also enjoyed your presentation, and hope that at some point we can connect, and then I can discuss more about the technologies that you're developing. So with that, I would like to end our e-seminar series today. This presentation, as we promised, and thanks to Shana, is going to be posted on our YouTube channel. So if you want to watch the videos again, please feel free to subscribe to our YouTube channel and then watch them online along with our other speakers' recorded videos. Don't forget to register for and attend our next speaker's talk. This is going to be on artificial intelligence. Again, this is our first AI talk, and it will be. I'm very excited to our next speaker's talk as well next week. So with that, thank you everyone for participating, and enjoy the rest of your day.
Thanks so much,
Anna.
Bye. Have a great day. Bye-bye.
Bye.